I'm Vignesh Vashankar, a junior electrical engineering student here at West Virginia University. I'm an undergraduate researcher at Dr. Mark Saitlin's lab at the biochemistry department here. Our lab specializes in novel EPR imaging systems for functional in vivo imaging. In this video, I will be presenting my work on the construction of a bioprinter from an open source 3D printer. The goal of this project is to design and create a 3D bioprinter that specializes in printing tissues with embedded probes. The probes will enable the chemical microenvironment of the printed tissues to be imaged using the electron paramagnetic resonance or EPR system. A bioprinter is a device which prints biological tissues and organs from a substrate using 3D printing techniques. The substrate is referred to as bioink and can take different forms. Various bioprinting strategies exist, each having its own bioink and polymerization mechanism. In all cases, bioinks contain biomaterials, proteins, and cells. Current applications of bioprinting are creating tissues and specific cells for in vitro testing. The expected future applications are the creation of replacement organs. I remodeled a commercially available 3D printer that prints with plastic to fit the requirements of a bioprinter. I replaced a heated plastic extruder with an automated syringe, which is made of a stepper actuator that pushes a syringe containing the bioink. This apparatus was mounted onto an aluminum frame and attached to the conveyor belt. The appropriate electrical connections were made between the actuator and the printer. The bed heating and extruder heating were turned off since they were not necessary and would damage the cells in the bioink. To integrate the new hardware, I made changes to the printer's firmware so that it can recognize and control the new setup. Changes in calibration, movements, and extrusions were achieved by editing the firmware. The first step in testing the capability of this newly constructed bioprinter was to ensure that it can effectively extrude a substance with similar viscoelastic properties compared to bioink. Hand cream was chosen for this test. A few designs like the one seen in picture A were made using the printer, which turned out as expected. We moved on to testing a couple of bioprinting strategies, photopolymerization, and freeform reversible embedding of suspended hydrogels or fresh. The spiral design in picture B was created using the photopolymerization process. The printed object was made out of synthetic resin and an EPR oxygen probe. The presence of the probe in the printed object was verified by a functional EPR image that is shown in picture C. The probe is distributed throughout the object, which is what was expected. Another object resembling a blood vessel was printed using the fresh method. The object was printed onto a gelatin medium in a petri dish. The outcome is seen in picture D. The printed object completely polymerized inside the liquid medium and turned out in the form of a single suspended solid. Our immediate next step is optimizing the syringe movements on the bed to create objects with more resolution and precision. We plan to introduce more syringes with greater degrees of freedom to the printer, which would allow the printing process to be more streamlined and efficient. To image the bioprinted objects using our EPR system, a high-speed data acquisition system consisting of an analog digital converter and a microcontroller that we recently developed will be integrated in the system. Through this endeavor, we've successfully created a functioning bioprinter from a rather inexpensive 3D printer and other inexpensive parts. We've shown that this bioprinter is capable of printing with bioinks that contain EPR probes. By doing so, we have monitored the chemical concentrations of the bioprinted constructs, which is the first time this has been done using EPR imaging. The anticipated long-term impact of this achievement is the removal of the need for live animal testing. This will contribute to greater progress in fields like drug testing and cancer research. Thank you for watching my presentation. If you have any questions, please leave them in the comment section.